title of her slides, and works for the US National Oceanic Center down in, is it Miami we work in? Or, again, said from the perspective of February in an English winter. So. She's basically passionate about coral reefs, and I think she's going to tell us something about that and then reflect on it a little bit in the light of her faith. So thank you very much, Margaret. Over to you.
Thank you so much. It really is an honor for me to be here. I've had the chance to speak with quite a few of you up to this point in the weekend, and it's just a treat for me to get to think about some of these broader faith and science issues that honestly I haven't been exposed to that much in my either scientific or faith walk. So it's been a great opportunity for me. My talk is going to take a little bit different perspective and focusing down on maybe a smaller portion of creation. And let's see if I can get the buttons right here. And in light of my own research and the part of creation that I understand the best, and just some personal reflections as a Christian that relate to that, as opposed to the much larger philosophical issues that we've been thinking about a little bit. So hopefully that will be a helpful perspective. By means of preamble, I just wanted to use this as a beginning. This is a quote from a Faraday, it's on their website, but I think it's very useful in this context in distinguishing creation as a theological term, acknowledging the dependence of all that exists on the authorship of the Creator, with evolution referring to our current understanding of how God has brought this into being. And so when I use the term creation, when I'm using it to refer to that body of creation, the things that have been created, from individual organisms to the full ecosystems in which they exist. And one of the first points that I'll make, that as an ecologist and as a Christian, I have always been struck with the sort of parallel vocabularies that are used in these two worlds to refer to very similar concepts. And so as an example, as I said, creation, ecologists call this organisms, ecosystems, communities, cycles, and this sort of thing. The Bible refers to creation being fruitful. And ecologists refer to the resilience of ecosystems, that is their natural capacity to regenerate themselves, to recover after disturbances. And these are very parallel concepts. The obvious one about brokenness and sin and fallenness. In ecology and conservation, obviously the issues of ecosystem degradation are extremely important and obvious. And I think that these have very parallel applications as we think about these things. Tending and caring for creation in the scripture, and the endeavors that we have in the empirical, scientific, or in the empirical world about conserving and restoring the function of these natural degraded ecosystems. So this has always been part of my background in terms of understanding these biblical concepts and the ecological concepts in parallel. So in light of Ruth's theme that she's given us to work with, I'm going to try to describe to you some of the aspects here, the aspects of wonder, the aesthetic beauty. We've been hearing about this in a lot of different contexts. But I think particularly coral reef ecosystems provide us an amazing example of complexity and of the function that comes out of that complexity as aspects from which we can draw wonder from God's action in creation. We'll touch a little bit on this brokenness, loss of function in terms of environmental degradation. I'll try to keep that to a minimum. A lot of coral reef talks go down a very sad road there. But then speak a little bit too from an important aspect of my research as we look at trying to restore these systems and what that looks like and what types of steps we're really thinking about in science related to restoration. So I guess I would suppose that what most people know about coral reefs, if they know anything, is that they are beautiful. That's, I think, the common knowledge about coral reefs. And it is indeed true. They're beautiful, I think, at several scales. Here's one from space. You can see the barrier reef surrounding this island. Sort of at the community or landscape scale, we enjoy colors and textures and many different creatures, but just the pure myriad number of creatures and variety that occur on coral reefs is truly dazzling. And honestly, this is a lot of where I became interested in coral reefs and desiring to study them for my career. There are also many aspects of complexity and function that coral reefs display. They do house extreme biological diversity. Interestingly, this diversity is much greater at higher levels of biological organization. So the level of phyla, coral reefs are actually the most diverse communities on Earth. They're not in terms of species, right? There's a lot more beetles in tropical rainforests, for example, but there are many more phyla of organisms that exist in marine and coral reef systems. Coral reefs have incredibly complex food webs, and they're based on some sort of unusual types of pathways, and we'll speak more about symbiosis in a moment. But this figure is one depiction, a very simplified one, obviously, of a coral reef food web. And food webs are very common concepts in ecology. Again, and so this relates to some of our discussions last night, and where I was describing this is a very common sort of depiction for ecologists of how ecosystems work, how energy flows through ecosystems. And I guess, part of this, in my mind, derives from the fact that creation is finite. We have a finite world, physical world that we live and exist in. And so these type of cycling and recycling of those physical substances are part of the necessary design in order for life to continue. And this is depicted with, the little guys here at the top, the plankton and so on, and the corals and the sunlight, and getting down to the larger predators here at the bottom. Now this food web doesn't depict people, but I would emphasize that people are a part of this, and that many people in societies derive a large portion of their protein directly from coral reef ecosystems in their environment. To a large extent, this diversity and complex food webs are enabled by the particular architectural complexity. So corals specifically build complex architectural structures, and this is largely what enables or supports the biodiversity and function that coral reefs display. And as I mentioned, again, a result of these complex functioning, there are very specific services that human societies derive from these systems as well. In addition to food, obviously tourism and recreation are important aspects of coral reefs. And also coastal protection. These architectural structures are important in protecting our coastlines and tropical areas from storms and erosion and this sort of thing. So I'll suggest that these two particularly important aspects of coral reef ecosystems are due to the corals themselves. So the types of critters that build these complex structures, and relatedly that display these complex symbioses. So this is not a parallel vocabulary thing, but this is just an analogy that I use. Now, when I'm speaking to school children in the US, I think their concept of a monster is much more related to Hollywood, but the actual classical definition of a monster being a combination critter, so a mythological creature that's composed of two completely dissimilar animals fused together. And so this is a concept that I think is helpful in understanding what corals are. Now again, the ecological science term that we use is holobiont. So corals are in fact a consortium of many, many different dissimilar types of creatures that reside together. So in the first case, this is just a picture of a surface of a coral. You can see the individual modules there. We call them polyps. You can see in that polyp an individual mouth in the center of each circle and the tentacles surrounding it. This is sort of the side view of that polyp. The coral skeleton is laid down here below that living tissue layer, which occurs on the top. But inside the coral tissues here, we have these little plant cells. We call them zooxanthellae is the name of those plant cells. But this partnership between a plant and an animal that are intimately connected is that partnership provides carbohydrates from photosynthesis that the coral enjoys as an export from the plants. And this subsidy of productivity of carbohydrates from the internal plant function inside the animal is largely what enables the calcification and the building of these architectural structures are largely due to this partnership between the plant and the animal. So this is a key aspect of reef building corals that enables the development of reef structures and these many complex interactions that they house. Now, so we've got this plant and animal monster, but it's actually, and you know, through the 70s and 80s, this was our sort of concept of how corals functioned and the important components. But as we have become more sophisticated in some of our genetics and other things, we're gaining a much greater understanding, as we are of humans, in fact, that there is a very rich community of microbes which reside within our bodies and are very important in regulating health and disease, and we're learning that the same is true of corals. So not only do they have these plant partners, which are intimately associated within their bodies, but they also have these immensely diverse communities of microbes that are crucial in their functioning. I'll just, again, this is a complicated figure, but this just shows, again, we can sequence all of these things now fairly easily. The diversity of life of just the microbes, which are found in an individual coral sample. And not only are there so many microbes, but they're actually specialized within these different tissue layers. Again, even though that tissue layer is quite small, some of these core microbes are associated with those plant endosymbionts. Some of them are specifically associated with the animal tissues. Some of them are very specifically associated with this surface mucus layer, which is on the outside of the coral tissue. So there's a huge diversity community in this holobiont of microbes, as well as the plant cells and the animal host, which constitute this entity which produces coral reefs. And as I said, we have a very small understanding, but we are gaining in that understanding that this complex association of microbes is crucial in regulating health and disease in corals. And we know that some of these microbes do specifically produce antibiotics within the coral surface. And we also have certain examples now where we can see that certain types of environmental stress, such as heat stress, and we'll talk about that more later, can cause imbalances in these microbial communities, resulting in health problems, mortality, death, those sorts of things. So there's another sort of fundamental aspect in which we can think of corals as communities themselves. So we've talked about them being this sort of consortium of different types of critters. As was mentioned, I work for a fisheries agency, and so many of my colleagues are involved quite diligently in stock assessment, in counting things, and understanding the status of populations of things like salmon to determine, well, how many salmon can we remove from that system and maintain a functional population, things like whales and turtles. And they're very well developed scientific methods for counting these things or estimating their abundances, and if we can know what their sizes are, often we can extrapolate lots of information about those populations, such as their birth rates and their death rates and their growth rates and their productivity. And this depends on us being able to count them. Well, that's one, and that's another one, and that's another one over there. And we sort of take that for granted, but in corals, this is a very complicated notion. So Santelises describes sort of three separate axes or characteristics upon which the concept of individuality of organisms is based. And these three are autonomy, all right? So I have an independent existence from the guy over there. I make my own decisions, my own physiological functions are happening separately. Genetic uniqueness, so I have a genetic makeup that's distinct from the guy over there. And then genetic homogeneity. So that means within my body, I have a single genetic identity, as opposed to a mixture of several. So corals, in fact, are challenged, can we say, in each of these three dimensions of individuality. We sort of mentioned before, this is sort of a cartoon depicting these sort of different levels of organization that coral populations exist in. So we mentioned the polyps or the modules that compose coral colonies. These modules have some variable degree of autonomy from the other polyps to which they're connected. And we don't understand this perfectly. We know that, as I said, they have a separate digestive tract. We know that they have separate reproductive capacity. But we also know that nutritional resources are shared amongst the polyps within a colony. So there's some in-between arrangement in terms of autonomy of modules within a colony here. Although in some sense the fitness element, the production of gametes, is performed at the polyp level. We have colonies, the colony unit of corals. And these are depictions of the Caribbean elkhorn coral. It's one of the species that much of my research is focused on. As a dumb field ecologist, when I go out counting things, these are usually the things that I can count, right? They're colonies of independent structures that are growing on the bottom. But particularly for branching types of corals like this, there is a very common occurrence of fragmentation. You can see as a branching morphology like this, often different types of physical disturbance can break a branch off, and those branches are quite capable of tumbling, next door, reattaching themselves and forming brand new colonies. So these species are highly clonal. So when I look at one colony and I look at a colony, next door, I have no way of knowing whether they're genetically different individuals or whether they're the same individual. And these species are highly clonal. And in fact, it was only about the past 10 years or so that we've had the genetic tools to even answer that question. Are these two colonies the same genetic individual, or are they different? And so this is obviously a fundamental question when we're trying to understand how many there are, or what is the status of a population. And so this depicts, and in this case of this species, we often have entire reef landscapes of happy, healthy-looking coral that in fact are monotypic. They're only a single genetic individual. And so when we're assessing the status of a species like this, and we may go out and observe hundreds and hundreds of colonies that look great and happy and everything is looking well, if we don't realize that they may be constituted by a single genetic individual, that the status of that species maybe is very different than it would appear. So this is a challenge to understanding the status of species. And then just last, I'll just mention this concept of genetic homogeneity. We heard about chimeras the other night, but corals are naturally subject to some degree of chimerism. We have studies now that show maybe about 2% of natural wild populations of corals are indeed composed of more than one separate genotype. And so this, again, further just complicates, well, what is it that we really should be counting when we're worried about coral status? All right, so I'm going to speak a little bit now about coral life cycles. This is an aspect of complex function that corals display that I've spent a fair amount of effort, or some would say effort beating my head against the wall trying to study and understand better. And in the years of experience that I have trying to study this, I will tell you that to a large extent I view the existence of any corals in the world miraculous, because the number of these steps and bottlenecks that corals go through in their life cycle is quite staggering. So, as I mentioned, as a dumb field ecologist, when I go out and try to study corals in nature, I can see these adults, and I can probably see a few of these kind of, younger, smaller colonies, but I can't really see anything that's smaller than a centimeter, even if I'm looking carefully on the reef surface. And so that means that the majority of the life cycle here is really a black box from an ecologist's point of view, or that we can't really directly observe. They happen at microscopic scales. These processes happen at night sometimes, and in the open ocean environment where we don't even know to look for them. So this is complications, and our knowledge is built rather slowly about these processes because they're difficult to study. I'll begin with the process of spawning. Many of the important reef-building species of corals are broadcast spawners. So this means that their gametes are shed into the open ocean. And so all of the steps of fertilization all the way through development have to happen. These guys have to fend for themselves in an open ocean environment. in order for broadcast spawners to be successful in producing larvae, they have to be very carefully synchronized. In other words, because eggs and sperm from different, and not just different colonies, but different genotypes are needed to have effective fertilization, that means that different genotypes that are close enough together spatially have to spawn within really 20-30 minutes of each other is our best understanding. They have to be that closely synchronized in order to have successful fertilization in the open ocean. And so if you think about that for a moment, I mean, that maybe doesn't sound too impressive, but if you think about it, a creature with no brains and no sensory organs that we can really identify somehow are able to calibrate within themselves 3 separate clocks. So this includes, a monthly clock. And so I guess I didn't say, the spawning only occurs 2 or 3 nights a year. So throughout the entire yearly cycle, these creatures have to keep track of a monthly or seasonal clock. They have to keep track of which night during the month it is. And then they have to keep track, somehow they have to figure out what time it is within that night in order, as I said, to synchronize by this, say, 20 or 30 minute sub-hour scale that this synchrony needs to occur. And so that's pretty amazing, I think. We know that they use temperature in terms, particularly this sort of seasonal scale clock. We know that they use lunar calendars. I mean, the way that we predict when these couple of nights per year are going to happen is based on a lunar scale. So, it's so many nights after the full moon in either August or September, depending on when the moon is in the month, and we're not usually sure which month they're counting from. It's likely they use day length signals. We sort of suspect that there's probably some chemical communication happening within populations at those finer, like maybe hourly or minute scales, but that hasn't really been well characterized. And perhaps it's not surprising that this doesn't always work. So the populations that I have studied carefully in the Florida Keys, this process often fails right from the get-go. So some years we don't observe any spawning at all within this time frame that we expect it to happen. On many other occasions, we might have only a single genet at one site that spawns on one night, say night 3 after the full moon, and 2 or 3 other genets at the same site spawn the next night or 2 nights after that. So they're not doing as good a job as we would hope they would do in this aspect of synchrony. Now, there's a couple of competing hypotheses why this might be. We could infer that there is some sort of environmental impediment, maybe some chemicals in the ocean or some sort of process that's impairing some aspect of one of these clocks. And so that sort of represents a breakdown in the natural process. An alternative hypothesis is simply that this is a species which used to be ubiquitous and hyperabundant in these reef habitats. And so it may just be that the selection pressures were never particularly strong for them to be on the same night. And so as long as they spawned within a 5 or 6 night window, there was somebody else nearby. And so the selection was just never strong. Now that this species is rare, the fact that that synchrony was never driven to be so precise is really a problem. So we can't distinguish between these two hypotheses. What we're left with is a lot of times we observe corals, these corals investing a lot of physiological resources in spawning with what we understand to be very little or nil outcome in terms of successful larvae, simply because of their timing. So the next stage in this process, you might recognize these guys. These are embryos from the talk the other night. These are coral embryos in a couple of different phases, and I'll point out these round ones actually are unfertilized eggs. So as I sort of alluded to, this process of fertilization is actually also quite problematic for these organisms. In our work, we're actually out there in the middle of the night, sort of putting tents over these corals to collect the gametes as they're released, and trying to keep them at high concentrations as opposed to letting them, drift off and dilute in the ocean, actually keeping them at higher concentrations. And we often still observe relatively low rates of fertilization in individual occasions where we're trying to help these corals along. And we've now been able to run actually more careful experiments that really suggest that individual parents, even ones that are spawning on the same night at the same reef, are not genetically compatible in terms of being able to have successful fertilization. So we see high variability in individual parent crossings, say between 10% fertilization all the way up to 80 or 90. So again, this suggests that even when we have multiple parents spawning on a reef, we may still not have very good larval production because by whatever mechanism, they're not able to successfully fertilize at this stage. So that's another point of concern. Again, so these guys, once they're fertilized, are in the open ocean, they're fending for themselves. It's obviously a hazardous environment, and many of them don't make it, of course, but the ones that do progress to this later phase. This is called a planula larva phase, and you can see it's got some shape. These this stage are able to swim a little bit. They can direct their motion. They have behavior where they can swim towards the bottom. Again, they're needing to find a home is their next step. Over here, I just wanted to give you, this is obviously under a microscope, but this is how their actual size. So they are barely visible to the naked eye. But these little simple critters have amazing sensory capacities, even though, as I said, they lack what we would identify as sensory organs. And they use this wide range of cues from the environment to make this very important decision that they have to make. That is, they have to find a spot on the reef where they're committed to spend the rest of their lives. They don't get a second chance, because once they, the process is a metamorphic process. So that planula larva that you saw in the previous picture undergoes a radical body transformation into a polyp. And that happens over the course of several hours. When it finds the spot it's going to choose, you can see the mouth and the small tentacle buds here on this very newly settled coral. Now, there are two things that this guy still doesn't have. He still doesn't have those plant symbionts, the zooxanthellae. With broadcast spawners, they don't inherit those from their moms or their dads. So that's a separate step that these minuscule critters have to somehow scavenge these partners. And again, we don't have any clue really how they do it. We know they pick them up. That infection seems to not be as limiting as I initially expected it to be. But that's an important step that still has to occur. And then within a couple of weeks, we'll begin to see the development of skeleton. That it kind of shows up as a sort of a white lacy structure that you can see within the tissue. That's that skeleton. And so then that's the beginning of the coral turning into the large structures that we observe as reefs. This photo down here was just sort of an illustration as we gain what to me is an amazing understanding of the sensory capacity. So some work that my graduate student did showed that corals prefer to settle on red substrates. So how on earth can a coral larva like that discern what's red and what's not red? And in fact, you can see here, we've seen quite a few images like this earlier of cells, but these, this is an image of one of those planula larvae, and the little red streaks there are that's a labeling for an opsin. I think we heard opsins referred to yesterday. They're photosensitive pigments that we have in our eye structures. They're universal across many visual systems. But coral larvae actually have specific opsins that are differentially expressed in cells on this aboral end. That's the butt end of the larva. That's the one that has to go and find a place to sit down on the reef when it metamorphoses. And in fact, there is this aggregation of light sensory cells, wavelength sensitive cells in coral larvae. And I think that's amazing. So as we think about these different processes, as I said, the conclusion that I come to is it's kind of miraculous that any corals exist in the world at all. And especially in a world where we have lots of different types of environmental changes going on, this complex life cycle means that corals are subjected and affected by a range of different types of threats within the environment. So the larvae we know are more sensitive to obviously water quality issues. After they settle, there's a host of competition and other types of interactions that happen on the reef bottom that are important and can impair their survivorship in these post-settlement phases. And our work with both elkhorn coral and some of the other diminished coral species in the Caribbean suggests that there really are aspects of depensation. And what we mean by that is there simply aren't enough parents around to successfully support and replenish populations of these species. So these are all aspects that we need to think about then as we think about, well, how do we help corals recover and be restored? Because I think we do have good evidence that this fruitfulness aspect of corals, their resilience, at least in some of these impaired Caribbean species, is already at a state where that impairment is such that fruitfulness or resilience is not operating in the way that we expect or hope it to. So, of course, and I'm sure you all know this and have heard these stories perhaps a lot, but there are many aspects of human activity that impair the fruitfulness of creation. And ocean ecosystems are no different. Corals appear to be particularly sensitive to some of these impairments, as I've described in terms of them having complex life cycles. But overfishing is important. We're changing the atmosphere. All due respect to my Norwegian colleagues, but climate change really represents a threat to an entire ecosystem, to an entire aspect of God's creation that possesses these characteristics of wonder and serving human needs and function and beauty. Warming is something that corals are particularly vulnerable to. Their optimum temperature, when they grow the best, when they're happiest and healthiest, is very close to their sort of upper thermal limit. It's within a couple of degrees. And so that means, we may be happy at a certain temperature, but we have tolerance for quite a ways above that. We're not going to kick off dead. Whereas corals tend to kick off dead within a couple of degrees of their optimum temperature. So this makes them particularly vulnerable. This graph here, this shows the sort of trajectory of abundance of elkhorn coral in the population in the Upper Florida Keys that we've been monitoring. This goes back to about 2004. I'll mention that in 2004, there had already been a lot of elkhorn coral lost already, but that's when our data set picks up. And you can see these massive collapses in population abundance. There was one that occurred in 2005. This was due to the occurrence of several hurricanes and the associated amount of what we call coral disease. What we mean by that is the corals up and died, sort of while we were watching them. There's a health aspect to that, but in using the term disease, it doesn't mean that we can say, oh, they had flu, or they had, it just means that they up and died. There was something that went wrong with their physiology and they died. And we see that as a major aspect of coral loss. It's often associated with other types of disturbances, such as in this case, it was associated with several hurricanes and physical disturbance. You see this other very recent collapse was associated with warm weather events, warm temperature stress, which we've experienced for the past 2 summers actually in the Florida Keys, and has resulted in extreme levels of coral mortality in very short periods of time. In between, you do see some recovery, right? This is a slightly upward slope, and so the coral can grow. And I'll mention this recovery that we observe is simply from the expansion of the remnant colonies that were there before. In this population, we have indeed observed 0 recruitment of new corals or new genotypes into this population from larvae. So this is just expansion of sort of remnants that we're able to observe. But clearly we appear to have this sort of return time of these mass disturbance events in the order of 7 to 10 years, as we've experienced in this region, relative to what would be an actual recovery time maybe of 15 to 20 years. So that of course doesn't set you up well in terms of the status of your species. Hence, as was mentioned, I work for the US government, and we have a law called the Endangered Species Act. And since about 2004, we have been asked to consider if corals, in fact, are at risk of extinction. And that's a rather daunting thing to be asked to determine. Partly, as I mentioned before, because we kind of don't know how to count them and we kind of don't know how to assess that in an objective scientific way, but also because it's a pretty uncomfortable thing to think about. The result of several of these processes, essentially we now have 20 species of reef-building corals that our government considers likely to become at risk of extinction in the foreseeable future. That's the legal language that our law attributes to this status of a threatened species. This photo just depicts these 7 here are the ones that reside in the Atlantic Caribbean region, so they're the ones that I deal with directly. The other 15 are in the Pacific region, the Indo-Pacific Ocean. And so there's a fundamental difference actually between these two types of, the two genres of species here. The ones in the Caribbean reefs are generally further down this trajectory of degradation than Indo-Pacific reefs. And that's reflected in the fact that 5 of these 7 Caribbean species that are listed are in fact the main species, the keystone species of Caribbean reef systems, as opposed to these specific species are mostly the rare species, the species within an ecosystem that we might expect just because they're naturally rarer to be more of an issue. So anyway, as part of the protections that this legal status carries, we're obligated to try to recover them, to fix them, to bring them back to a state where they have this capacity to regenerate. And so one of the actions that's been identified is population enhancement. We're trying to restore populations by, in fact, the term is coral gardening. This is a term that's in the scientific literature. And this is a daunting prospect, given all that we don't know about coral genetics, about coral health and disease. But we've pressed forward developing a broad network of coral nurseries. We can grow a lot of coral. This is one of the aspects of research also I'm involved in. This just shows an example of some of the things that we're hoping to learn to improve the outcomes of this endeavor. This is an experiment. This is kind of hard to see, but there's 4 fragments of elkhorn coral here. During the warm event that I mentioned we had last summer, these are different genotypes, and this one bleached consistently badly, and this one down here actually didn't. So we are able to begin seeing differential resistance or different traits that different genotypes have that we're hoping to use more effectively in restoration. So essentially we're at a stage in coral reef ecology or restoration where we're contemplating sort of reverse engineering these ecological systems to restore the resilience that's been lost. And I find this a very humbling endeavor. I sort of draw hope in my understanding as a Christian that God cares about these ecosystems and cares about them functioning properly. And so that it aids me in walking forward in this path. There are those, and there is a scientific discussion that's initiating now, which kind of goes a step further than that. The term assisted evolution has been introduced in the literature now with regard to corals, rising from this recognition that the environment, specifically the climate change environment, the temperature environment that corals live in, is changing faster than the natural evolutionary processes have an ability to adapt to. And so if we want corals to persist, if we want these functioning systems to persist into the future, there may be some more meddling that's going to be required in their biology. And there's a continuum here of actions, obviously, but the idea at this stage is that we need to be developing these scientific tools, understanding what the potential of some of these tools, again, and so the ideas are here where we have these consortium of symbiotic partners. Maybe we can tweak these partnerships in ways that could increase their resilience or their adaptability, their ability to survive in warmer temperatures. Or maybe there are certain types of selective breeding if we can identify these traits in different organisms. So this is an active area of discussion now that we need to be doing science on this. We need to be understanding what the potential of some of these more meddling approaches might be if we want to ensure the function of coral reefs into the future. So this is the fundamental problem. We can grow a lot of corals actually. Reconstituting them into fully functioning ecosystems is something we still really don't know how to do, but we're needing to learn quickly, and that's kind of the place that my research resides at the moment. So real quickly, because I need to finish up, but as I was putting this talk together before, I learned a lot of the things that I've learned this weekend. I guess what constitutes natural theology, I had taken it from the Romans passage that was quoted last night, but this certainly is an aspect. Understanding, as I learn more about the complex functioning of coral reefs, I certainly see God's, his beauty, his desire for us to have mutual sustenance manifest in that. And I view a large aspect of my research tending towards or hoping to support the carrying and tending for creation in order that that functioning can persist into the future. And my reverence and the urgency in pursuing these things are reinforced by my faith. I'll just circle back to beauty really quickly. This is a paper that came out really recently where they actually quantified the beauty of individual images, and they can do this with machine learning algorithms now. And in looking at images of different types of coral reef systems, there is a very strong correlation with an independent measure of those coral reefs' functional capacity or the degree of impairment that they have from human impacts. And so I think that this is, I mean, maybe that's sort of a no duh, but I think it's pretty interesting that this is an objective characteristic of coral reefs. I'm going to end with a quote as well, and I don't have it written here. My quote isn't from a philosopher or a theologian. It's actually from a fellow coral reef ecologist. His name is Peter Sale. He's retired, and so he has the opportunity to think more about some of these sorts of things. But I'm going to close with a quote from him. It says, "The more that we learn about this amazing place and its amazing life forms, the more intricately complex and absolutely improbable it seems to be." I'm not aware if he's a Christian or not. I presume not. Perhaps that perspective can help some others of us embrace the idea that our only home, this tiny blue marble with its living, enveloping biosphere, has to be respected and cared for, and that such respect and care should be a prime mover in our lives. Thank you.
Thank you so much, Margaret. You opened our eyes to a bit of this world not many of us get the chance to look at. So let's throw it open for some questions and discussion. Alexander.
Thank you for this fascinating talk. I didn't know much about coral reefs before, but it's amazing. What I just know from a quiz is the question, what is the largest living organism on this planet? And the answer was the Great Barrier Reef. When I heard your talk, I thought it's not that easy. Is the Great Barrier Reef one organism, or is it composed of different organisms? What do you think about that?
Yeah, that might be a bit of a trick question. I mean, I would not consider the Great Barrier Reef as a single organism. You know, it's an interacting biological system, certainly. And, you know, I've heard I think the variant of that, it's, you know, there's only like 2 or 3 structures that are visible from outer space. And one of them is like the Great Wall of China is the only like human built one, but the Barrier Reef in Australia is this, you know, massive structure that's visible from space. So it speaks to the sort of significance of what these little teeny organisms are able to do. But I would agree with you, I probably wouldn't say that the Great Barrier Reef is a single organism.
Hi, thank you so much. That was a really, really interesting talk. What's the significance, do you think, wider significance of the fact that it's jolly so hard for these things to reproduce? What does that mean?
What does that mean? I struggle with that as well. I probably don't have a good answer to that. One tendency is to think, well, wow, we've already, there's already been so much deterioration in how this system is supposed to work. The concept is these organisms spawn in such abundance. There's such a gajillions, of larvae that should be out there that adequate numbers are able to recruit. And the other thing that we know about these types of species, especially these big reef-building species, I think Jeff had mentioned last night, are selected and K-selected species. So we understand that the biology of these species is that they don't recruit very often. They grow for a long time, they grow very large, they reproduce, they go through many reproductive seasons, but the successful reproductive events may be very rare. But that's the strategy that those species are built upon. Now, the problem that we have now is that the rates of mortality, right? The rates of mortality are supposed to be very low in those species because they live a long time. Right now, we're observing these species to die very rapidly. And so that basic ecological strategy isn't working, even though it may be that they're not supposed to reproduce, but every 100 years maybe. But when you're dying this fast, that strategy isn't working for you. So both the mortality processes and the recruitment processes obviously have to be fit together in a successful strategy.
And is it true that actually coral is much more, one of the most vulnerable ecosystems to things like pollution and acidification and so on and so forth?
There are quite a few aspects of coral reefs that do make them particularly vulnerable. So in terms of pollution, coral reefs exist in shallow coastal systems, so they tend to be in close proximity to lots of people. And so that tends to increase their exposure to lots of types of pollution. I mentioned their temperature sensitivity, and this is another aspect which makes them particularly sensitive because they have such a sensitivity to thermal extremes, even very small thermal extremes, to which they can die very quickly. And you mentioned ocean acidification. I didn't go into that really, but because calcification is such an important aspect of coral biology, this is the process we understand is most vulnerable to changes in ocean chemistry, which we refer to as ocean acidification. So they manifest this sensitivity, particularly to these chemical changes in the ocean as well. And I might add, some of these reproductive processes and early life stages, we understand, are probably most vulnerable to those chemical changes of ocean acidification. So the fertilization step that I mentioned is kind of tricky. At lower pH, we've got even more problems in terms of a successful fertilization. And that process after they settle, where they begin making skeleton, that's the other phase, which we have building evidence is very vulnerable to these changes in pH as well. So yeah, those three, I would say they do have an unusual susceptibility.
Jeff, you had a question and then we'll come up.
So Margaret, there has to be a really great fossil record of corals, right? So
Absolutely.
what's known about cyclical extinction and sort of bottlenecks with coral, and is there something unique about current environmental conditions that have never been seen in the history of the planet, or can you address some of that?
Unfortunately, I cannot address it well. You're right that corals have an excellent fossil record and there is a lot known, and I am not going to be able to answer that well. However, the types of species collapse. So, for example, this acropora, this elkhorn coral, is one that shows it very clearly in the fossil record. It can be identified. And we know that there have been collapses in these species in the past, but they have occurred at a different time scale. So what we observe now is happening at a much more rapid time scale than the changes in the past. Corals, there have been mass extinctions. Corals have persisted over a very long geological time, but unfortunately I can't put the numbers on off the top of my head. So I can't give a good answer to that, I'm afraid.
There was a question here, yeah. And then
Thank you. You mentioned machine assessment of beauty.
Mm-hmm.
Can you elaborate on that? Can it work for things other than corals?
Well, I believe it can. My understanding is these algorithms were derived from human assessments of art, right? So paintings and things like that. And this part of the work was not done by coral reef ecologists. There were other algorithmic computer scientists, I think, that took scales and particular visual aspects about color and contrast and texture and these types of image characteristics that do predict apparently human perception of beauty in human produced artworks. And that was the type of approach that was used in that study, developing, as I said, computer algorithms and machine learning for those characteristics, and then assigning these sort of beauty scores to the images of coral reefs. But yes, those are algorithm characteristics derived from, I guess, psychology or art or whatever other aspect of human endeavor that the coral reef ecologists borrowed.
So ultimately it depends on human assessment.
Exactly.
Yeah.
It's the human perception that's embodied in those algorithms.
Thank you.
There's a question here.
Thank you for an interesting speech. This thought of like helping corals to survive clearly resonates with us. I would like to take a kind of devil's advocate view of that, like ethically. Do they deserve that we help them more than other people or other things that we could help? And so are we to help corals survive because they are beautiful? Are we also to make things survive that are ugly or evil?
Yeah, absolutely. So that's a really important point. I was not meaning to suggest that the beauty is the basis upon which that stewardship should be applied. I guess you raise really important questions because there are many needs in our world, many aspects of failed stewardship that we could invest in trying to fix. I guess I don't know that I can make an argument that coral reefs are more important than any of those other aspects. I guess I would say that I think it's very clear that doing a better job or exercising better stewardship in areas like climate change has equal benefits for people, the least of these in many senses, as it does for the natural world. I think in many cases, as I mentioned, there are many human communities that are intimately dependent on the coral reef resources in their neighborhood. And so I think there is very strong linkage between human livelihood and human well-being and this functional capacity of natural systems. I think that's true of coral reefs as it is of many different types of natural systems. Now, if we're at a point of distinguishing or prioritizing between coral reef systems and forests and other types of natural systems, that's obviously problematic, and at some level there may be, there's obviously resource limitations, maybe some of those decisions need to be made. But I would view, and I think there's a lot of evidence that, as I said, human well-being and the proper functioning of ecological systems are very closely linked. And so they need to be addressed together and by some of the same strategies. I think that's what I view as this sort of mutual sustenance in the design of the world that we live in.
Thank you very much for your talk. I've got two questions. The first is more of an observation. Someone yesterday was saying that organisms which are short-lived and fast reproducing adapt most quickly. Hope I've got that right. And therefore, something that's more long-lived and difficulty reproducing is going to adapt least quickly. So I would seem to generate a significant challenge. I just wondered if that was correct. The second question is, David Attenborough was presenting something the other day on British television. I hope I've got this right because I'm not a biologist, so please forgive my errors. I think the phrase was ARK projects or Noah's Ark projects, where there'd been some success with taking, I think, birds and some animals out of their natural environment, put them in a controlled environment for a number of years. This is for a species about to go extinct, by the way. And they've had some success in doing that and then releasing them back into their natural environment. Now notice your growing of the coral is in its own environment. I wonder if there's any possibility of taking it out of its natural environment, put it in a controlled environment, and then somehow beef it up, if that's the right word, And would it transplant back into the natural environment? Because you seem to have a problem, even in the natural environment, getting it to transplant from growth to actually a sustaining coral reef.
Those are really insightful questions. So I didn't have a chance to go into all of this. So there's a couple questions there. So the first one, it is true that short-lived, fast-growing species adapt more quickly, and it's just because they fit more generations in. And so whatever those adaptive processes genetically happen more rapidly. Now, on my assisted evolution slide, that's actually maybe one of the advantages that corals have, or one of the strategies that we may be able to use to sort of jumpstart them, because they have these important partnerships with small unicellular R-selected species like plant cells and microbes. And so one of the ideas is we may be able to use the greater adaptive capacity of those partners to improve the resilience of the holobiont, of the organism as a whole. So that's maybe an advantage that corals have over other less, intimately symbiotic creatures. So that's one point. In terms of the strategies for this sort of culturing and enhancement, most of the activities that we have going on now in the Caribbean in terms of culturing and outplanting corals, there's been a very precise and deliberate decision to emphasize in situ culture. And the reason is, as I alluded to, all the stuff we don't know. You can imagine when you take an organism out of its natural environment and you put it in a tank or a lab or a cage, that's a huge selective event, right? You're radically changing its environment. We expect that to have genetic consequences, right? That's selection, that's a huge selective event that you're doing. Then we take it from ultimately you take it from that land-based environment, you want to put it back out into nature, that's another huge selective event. So you're imposing two huge selective events. We don't know what the consequences of that are, but they worry us. So our strategy has been really to emphasize in situ culture, because in the interest of minimizing those differences. Now, I will say too, though, that there are efforts with corals as well to develop ex situ culture strategies as well. It's serving a little bit different purpose. It's obviously insulated a little bit better. When we can control the environment, they're not subject to the bleaching events and the storms that our in situ nurseries are subject to. So there's a separate purpose that ex situ culture serves in terms of protecting and sort of ensuring the persistence, but we're really uncomfortable with the idea of using land-based culture as the main propagation for enhancing or restructuring wild populations, just because of all that we don't know. In terms of, maybe they're developing some sort of microbial consortia when they're under artificial lights, that when you put it back out in nature is going to have one of these unintended consequences in terms of, worsening disease, because you're introducing something from an artificial environment. So we've moved forward quite rapidly in this endeavor of culturing and outplanting corals, but we've made deliberate decisions to do it in the way that we felt was less risky based on basic conservation biology principles. Does that help answer your question? But yeah, no, those are very perceptive points, and they're things that we worry about a lot in terms of how do we ensure the raw material to work with in terms of these species, but enabling and easing and making it the most effective and introducing the least risk in terms of reformulating them as natural populations.
Could I just clarify one point then? Because on one of your slides where you showed, I think, gardening, it looked like there were sort of bits of coral just sort of grown on a structure. Will those actually eventually be able to root, if that's the right word, in the ocean floor? Or
Yeah, so I didn't explain that process real well, but yes. So they're in the field nurseries for the most part. They're grown on those big floating structures. They grow quite fast and effectively. Those then are taken, those fragments are taken from the structures, which are in near shore specific nursery environments, and we paste them back out on the reef. Actually, let me just go back real quick. Maybe. So here they are growing under, and then doesn't show up real well, but then we take those fragments and we essentially glue them back onto the reef substrate in the reef environment where we're trying to restore them. So it's a two-step process: culturing them and then taking them from the nursery environment, which I said is this sort of intermediately artificial environment, and reintroducing them to the natural environment.
What's the difference? Sorry. What is the difference between assisted evolution and breeding?
So that's a really good question. Selective breeding is one aspect of assisted evolution within this terminology. So this is obviously something humans have done with organisms, you know, from long aspects of history. Now, I will say, so humans have done this for a long time, but they've done it in primarily in context of agriculture, domestication, right? Selective breeding hasn't really been done in the context of conservation as much. Now, in small populations, yes, right? The pandas, they have stud books and they know exactly who, you know, what the best strategies are for breeding those very small populations of mammals. But there's, sorry, I just lost my train of thought. Selective breeding is an aspect that we refer to under this umbrella of assisted evolution. It's one of several strategies. It's viewed rather more radically in the context of conservation than it is in other areas of human-animal interaction in terms of domestication and agriculture.
I guess it's subject to the law of unintended consequences.
Absolutely, that's the huge concern.
Because you don't know as much about a wild system as a
And the consequences may be, you know, our consequences in agricultural systems are still bad, right? Any, you know, our mad cow sort of thing, you see these unintended events that can cause a lot of problems. The concern, of course, is in a natural, uncontrolled environment. Those problems could manifest in much worse ways.
Any more questions for Margaret?
How often do you dive?
Well, a lot.
We're
Not,
about to get all really jealous.
I should say it's not as much fun and glamorous as you think, you know, as the guys on TV, because usually the weather's bad and, you know, usually it's cold and you have snot coming out of your nose and everything. So it's not quite as much fun as you might think. I don't dive as much as I used to when I was young. It's also hard on your back. But I do a lot of diving. That's where most of our data is collected in the ocean and, you know, working with the corals directly. So that is a major aspect of the interaction that I get to have with God's creation, and I'm really thankful for that.
Great. Well, let's thank Margaret again for that fascinating